Hey everyone, I'm going to continue on a couple of the videos that are going to break down the fates of pyruvate. And in this first one, we're going to talk about what happens to pyruvate when oxygen is present. And this would occur in eukaryotic cells as well as all yeast and bacteria cells. So when oxygen becomes present, the pyruvate that's formed in glycolysis that's out in the cytoplasm, that ultimately has to get into the mitochondria. And so what will happen, let me just draw the structure of pyruvate here. And this is in the cytoplasm. This is the matrix of the mitochondria. What will happen is that on the inner membrane of the mitochondria, pyruvate will then encounter this very large enzyme complex called the PDC, pyruvate dehydrogenase complex. So that's right there. And as pyruvate passes through this, the following reaction is going to occur. So pyruvate is going to ultimately be converted into acetyl-CoA. Okay, but along the way, there's going to be a couple other things that will happen. Notice that the carbons of the acetyl-CoA here are missing something from pyruvate. This first carbon of pyruvate is ultimately going to come off as CO2. So that's going to be liberated as a gas. And that's one of the reasons why I've shown this essentially as a one-way process. It's not really reversible because the carbon dioxide that's liberated just diffuses out as a gas. The pyruvate dehydrogenase complex also will produce an NADH along the pathway here. Now this enzyme complex is a very large enzyme complex. It's embedded in the membrane here. The three-dimensional structure actually kind of reminds me of something you might be familiar with. If you've ever watched the movie Star Wars, you remember the Death Star complex, or the Death Star that's there. I like to make the analogy that the PDC is kind of like the Death Star enzyme. It's a very large, multi-layered complex. Pyruvate floats in, and there's several enzymes that are in this complex that ultimately spit out acetyl-CoA. A couple other features you probably should know about this. Ultimately, it requires a very important vitamin. So it requires vitamin B1, which is also named thiamine. Okay, so there's an absolute requirement in our diets for thiamine, or vitamin B1. This enzyme absolutely requires that vitamin. And if you don't have that vitamin, you can't ultimately convert pyruvate into acetyl-CoA and ultimately make lots of ATP. Okay, so the pyruvate dehydrogenase complex, this is sort of the fate of pyruvate when oxygen is present. And the reason for that presence of oxygen is that ultimately acetyl-CoA is going to go to the Krebs cycle. And ultimately the electron transport chain, and then ultimately oxidative phosphorylation. Okay, and ultimately it's at this very end in the electron transport chain where the oxygen will be used. That's all happening inside the matrix. So you definitely would want to have a picture in your mind of where this is happening. Pyruvate dehydrogenase, again, is on the inner membrane of the mitochondria. The pyruvate that's made from glycolysis, we'll just put that here. So those 10 steps, this is glycolysis. This is happening out in the cytoplasm. And then ultimately, through this pathway, we make the key compound acetyl-CoA. All right, so that covers the fate of pyruvate when oxygen is present.